Euchromatin, CSIR Net Life Sciences High-Level Lecture. Euchromatin is a less condensed, transcriptionally active form of chromatin that plays a central role in gene expression and cellular function. Unlike heterochromatin, which is tightly packed and transcriptionally inactive, euchromatin exists in a more relaxed, open state that allows the transcriptional machinery, including RNA polymerase, to access the DNA template. Euchromatin is typically found in the central regions of chromosomes and contains genes that are actively transcribed in various cell types. The physical structure of euchromatin is dynamic, undergoing compaction and decompaction during the cell cycle, particularly during interphase, to facilitate or inhibit gene expression depending on the cell's needs. The primary structural feature of euchromatin is its loose, uncoiled conformation, which makes it more accessible to DNA-binding proteins, transcription factors, and RNA polymerase II. This open chromatin structure is achieved through specific modifications to histones and DNA, collectively known as the epigenetic code. Histone acetylation, especially at lysine residues on histones H3 and H4, is a prominent mark of euchromatin. Acetylated histones neutralize the positive charge of histones, weakening their interaction with the negatively charged DNA, thus promoting chromatin decompaction and allowing access to the transcriptional machinery. In addition, histone methylation such as H3K4me3, a mark of active transcription, and the presence of H3K36me3 associated with elongating RNA polymerase further enhance the transcriptional activity of euchromatin. Euchromatin is generally rich in genes involved in various cellular functions, including housekeeping genes that maintain basic cellular processes like metabolism, DNA repair, and cell cycle regulation. These genes are transcribed into messenger RNA, mRNA, and then translated into proteins essential for cell survival. The regions of euchromatin also often include regulatory elements, such as enhancers, promoters, and silencers, which bind transcription factors and co-regulators to finely tune gene expression. The active nature of euchromatin makes it a key player in cellular differentiation, development, and response to environmental stimuli. In eukaryotic organisms, euchromatin is also found in regions of the genome involved in immune response and stress adaptation, highlighting its role in dynamic gene regulation. Unlike the more rigid heterochromatin, euchromatin undergoes structural transitions during various stages of the cell cycle. In the G1 phase, euchromatin is largely decondensed, allowing for active transcription and DNA replication. During the S phase, some regions of euchromatin undergo localized condensation, while maintaining enough accessibility for DNA replication machinery. In the G2 and M phases, euchromatin remains compacted compared to its active state in G1, but it still remains more accessible than heterochromatin. This dynamic regulation of chromatin structure is crucial for maintaining the balance between gene expression and chromosomal stability. Euchromatin's dynamic nature is also regulated by various non-coding RNAs, including long non-coding RNAs, ln-RNAs, and small RNA molecules, which help modulate gene expression and maintain chromatin structure. These non-coding RNAs participate in the regulation of epigenetic modifications and coordinate chromatin remodeling complexes, ensuring the fine-tuned transcriptional output required for cellular functions. For example, the XISTRNA, responsible for X chromosome inactivation, inactivates one of the X chromosomes in female mammals by promoting the formation of heterochromatin. However, euchromatin remains largely unaffected by this process, underscoring the differential regulation of chromatin states in response to developmental and cellular signals. Euchromatin's role extends beyond gene expression. It is also essential for maintaining genomic integrity. Active transcription involves the generation of RNA, which can lead to DNA damage, particularly in highly transcribed regions of the genome. Euchromatin undergoes constant surveillance by various repair mechanisms, including nucleotide excision repair (NER) and homologous recombination, to prevent the accumulation of mutations and ensure the stability of transcribed genes. The ability of euchromatin to undergo chromatin remodeling also allows cells to respond to environmental cues, such as stress, by activating specific gene pathways necessary for survival. In conclusion, euchromatin is a vital, transcriptionally active chromatin state that plays a central role in gene expression, cellular function, and maintaining genomic integrity. Through its dynamic structure and regulation, it enables the fine-tuned transcriptional output necessary for cellular differentiation, development, and response to external signals. The modification and regulation of euchromatin are critical for maintaining cellular homeostasis and preventing disease, especially in the context of cancer and aging, where dysregulation of euchromatin structure can lead to epigenetic misregulation, gene silencing, or inappropriate activation of genes. CSIR Net previous year questions. Euchromatin CSIR Net Deck 2017. Which of the following histone modifications is typically associated with euchromatin? A. H3K9me3. B. H3K4me3. C. H3K27me3. D. H3K36me3. Answer. B. H3K4me3. Explanation. H3K4me3 is a hallmark of active euchromatin, associated with actively transcribed regions. CSIR Net June 2015. Euchromatin is characterized by a compact, transcriptionally inactive state B loose, transcriptionally active state C, rich in repetitive DNA D, highly methylated DNA. Answer. B loose, transcriptionally active state. Explanation. Euchromatin is open, less condensed, and actively involved in transcription. CSIR Net Deck 2019. Which of the following is a feature of euchromatin? A. It is rich in transposons. B. It is tightly packed during interphase. C. It contains regions actively transcribed into RNA. D. It remains inactive in all stages of the cell cycle. Answer. C. It contains regions actively transcribed into RNA. Explanation. Euchromatin is a region where active transcription takes place, allowing gene expression. This lecture provides you with in-depth, advanced content tailored to the CSIR Net Life Sciences exam. Let me know if you want the next topic, e.g., histone code, DNA methylation, etc. To continue your study plan. You said, transposons. ChatGPT said. Transposons, CSIR Net Life Sciences High-Level Lecture. Transposons, also known as jumping genes, are DNA sequences that have the ability to move from one location to another within the genome. They are a key component of the genomic plasticity of eukaryotic and prokaryotic organisms. Transposons can cause genetic variation, genomic rearrangements, and mutations, contributing significantly to evolution and the regulation of gene expression. The movement of transposons within a genome can be either autonomous or non-autonomous, depending on whether they encode the enzymes required for their own transposition or rely on the transposition machinery encoded by other transposons. There are two main classes of transposons, class 1 retrotransposons and class 2 DNA transposons. Both types play essential roles in shaping the genome, but they operate through distinct mechanisms. Class 1 transposons, retrotransposons. Retrotransposons move within the genome by RNA intermediate. Their transposition mechanism is similar to the process of retroviral replication. Retrotransposons first transcribe their DNA into RNA, which is then reverse transcribed into complementary DNA, cDNA, by the enzyme reverse transcriptase. This cDNA is inserted into a new genomic location, typically somewhere else in the genome. The major types of retrotransposons include long-terminal repeat LTR retrotransposons, non-LTR retrotransposons, and signed short interspersed nuclear elements. LTR retrotransposons are similar to retroviruses and contain long repeated sequences at both ends, LTRs. These transposons typically encode reverse transcriptase, integrase, and sometimes additional proteins involved in transposition. Non-LTR retrotransposons do not have LTRs and are classified into lines, long interspersed nuclear elements, and signs. Lines encode reverse transcriptase and endonuclease, which facilitate the integration of transposons into the genome. Signs are shorter, non-autonomous transposons that rely on lines for their movement. The retrotransposons can lead to insertional mutations, disrupting genes or regulatory regions, which can cause diseases, including cancer and genetic disorders. However, they also contribute to genetic diversity and can influence the evolution of gene families. Class 2 transposons, DNA transposons. Class 2 transposons, also called DNA transposons, move directly as DNA. They do not require an RNA intermediate like retrotransposons. The mechanism of transposition is mediated by the enzyme transposase, which is encoded by the transposon itself. Transposase catalyzes the cutting and pasting of the transposon DNA from one location to another within the genome. This process involves the creation of single-stranded cuts at the transposon's ends and insertion of the transposon into a new location, often causing duplication of flanking sequences. DNA transposons can be divided into autonomous and non-autonomous elements. Autonomous transposons carry all the necessary sequences for their transposition, including the transposase gene. Non-autonomous transposons lack this gene and depend on the transposase provided by other autonomous transposons in the genome. DNA transposons are often found in bacterial genomes, where they can confer antibiotic resistance through horizontal gene transfer. They are also present in the genomes of higher eukaryotes, such as humans, where they play a role in genomic evolution and genetic diversity. However, they can also cause harmful mutations and instability by inserting into critical regions of the genome. Transposons in genomic evolution. Transposons are often referred to as the selfish genes because they replicate and propagate themselves without necessarily providing a benefit to the host organism. Despite their selfish nature, they play a significant role in evolution. By causing insertions, deletions, and duplications of DNA, they create genetic diversity, which may lead to new traits or adaptations over evolutionary timescales. In some cases, transposons have even contributed to the development of new genes through processes such as exon shuffling and the evolution of new regulatory regions. Additionally, transposons can affect genomic imprinting and the regulation of gene expression by inserting into promoters, enhancers, or other regulatory regions. This can lead to epigenetic changes, influencing the phenotype of the organism without altering the underlying DNA sequence. Transposons also play a role in the structural rearrangement of chromosomes, influencing processes such as chromosomal breakage and recombination, which are important in meiosis and the formation of genetic diversity in populations. Impact of transposons on human health. While transposons contribute to genetic diversity, their uncontrolled activity can lead to genetic instability and disease. The insertion of transposons into critical genes or regulatory regions can result in mutations that cause various disorders, including cancer. For example, Lyme-1 retrotransposons have been shown to integrate into the human genome and disrupt tumor suppressor genes, contributing to the development of cancers. Moreover, certain diseases like hemophilia and muscular dystrophy are associated with mutations caused by transposons. In some cases, the movement of transposons can result in antibiotic resistance in bacteria, as they can carry genes that confer resistance to antibiotics, allowing bacteria to survive in the presence of drugs. CSIRNet previous year questions. Transposons. CSIRNet June 2017. Which of the following is not a characteristic feature of retrotransposons? A. They move by DNA intermediate. B. They move by RNA intermediate. C. They are present in both prokaryotes and eukaryotes. D. They can be classified into lines and signs. Answer. A. They move by DNA intermediate. Explanation. Retrotransposons move via an RNA intermediate, unlike DNA transposons. CSIRNet Deck 2018 DNA transposons require the enzyme, a reverse transcriptase B transposase C, ligase D RNA polymerase. Answer. B transposase. Explanation. Transposase is the enzyme required for the movement of DNA transposons. CSIRNet June 2016. Which of the following statements is true regarding the movement of retrotransposons? A. They replicate through an RNA intermediate and integrate as DNA. B. They replicate through a DNA intermediate and integrate as RNA. C. They use transposase for transposition. D. They do not integrate into the host genome. Answer. A. They replicate through an RNA intermediate and integrate as DNA. Explanation. Retrotransposons use an RNA intermediate, which is reverse transcribed into DNA for integration into the genome. This comprehensive lecture on transposons provides the required depth to address high-level CSIRNet life sciences questions. Please let me know if you'd like further elaboration on other topics or need additional questions and explanations. Photosynthesis is a fundamental process in plants, algae, and some bacteria, where light energy is converted into chemical energy stored in glucose molecules. The process occurs primarily in the chloroplasts of plant cells, involving two major phases: the light-dependent reactions and the Calvin cycle light-independent reactions. The light-dependent reactions take place in the thylakoid membranes and require sunlight to split water molecules photolysis, producing oxygen, ATP, and NADPH. These products are then used in the Calvin cycle, which occurs in the stroma of the chloroplasts, to convert carbon dioxide into glucose through a series of enzyme-mediated steps, most notably catalyzed by the enzyme riboscope-ribulose bisphosphate carboxylase oxygenase. In aerobic organisms, aerobic metabolism refers to the process by which cells extract energy from glucose in the presence of oxygen. This process is more efficient than anaerobic metabolism, producing a large amount of ATP. Aerobic respiration occurs in three main stages: glycolysis, the citric acid cycle, Krebs cycle, and oxidative phosphorylation, electron transport chain, and chemiosmosis. Glycolysis, occurring in the cytoplasm, breaks down one molecule of glucose into two molecules of pyruvate, generating a small amount of ATP. In the mitochondria, pyruvate is further oxidized during the citric acid cycle, releasing high-energy electrons. These electrons are passed through the electron transport chain, driving the production of ATP through oxidative phosphorylation. Oxygen acts as the final electron acceptor, forming water as a byproduct. The relationship between photosynthesis and aerobic metabolism is crucial for life on Earth. Oxygen, produced during photosynthesis, is essential for aerobic respiration, while the carbon dioxide produced by respiration is used in photosynthesis. This cyclic relationship sustains life by providing energy and maintaining atmospheric oxygen levels. Key concepts to focus on. Photosynthesis. Light-dependent reactions, occurs in thylakoid membranes, splits water to release oxygen. Calvin cycle, converts CO into glucose in the stroma using ATP and NADPH. Role of riboscope in carbon fixation. Aerobic metabolism. Glycolysis, occurs in the cytoplasm, producing pyruvate and ATP. Citric acid cycle, takes place in the mitochondria, generating high-energy electrons. Oxidative phosphorylation, involves the electron transport chain and ATP synthesis, utilizing oxygen. Interrelationship between photosynthesis and aerobic metabolism. Oxygen production in photosynthesis supports aerobic respiration. Carbon dioxide produced in respiration is used in photosynthesis. Previous year questions. Question CSIRNet 2019. Which of the following statements about the Calvin cycle is correct? A. The Calvin cycle takes place in the mitochondria. B. ATP and NADPH are consumed in the Calvin cycle. C. The Calvin cycle uses oxygen to produce glucose. D. The Calvin cycle produces carbon dioxide as a byproduct. Answer. B. ATP and NADPH are consumed in the Calvin cycle. Question CSIRNet 2021. In the process of aerobic respiration, which of the following steps produces the most ATP? A. Glycolysis. B. Citric acid cycle. C. Electron transport chain. D. Fermentation. Answer. C. Electron transport chain. Question CSIRNet 2018. During the process of photosynthesis, the ultimate source of oxygen released is from which of the following? A. Water. B. Carbon dioxide. C. Glucose. D. Oxygen. Answer. A. Water. This lecture provides you with a thorough understanding of photosynthesis and aerobic metabolism, focusing on the key processes and concepts essential for CSIRNet JRF life sciences. The inclusion of previous year questions helps reinforce your preparation and understanding of the topics. Let me know if you'd like me to continue with another topic or make adjustments. You said. Cell division and cell cycle. Mitosis. ChatGPT said. Cell division and cell cycle. Mitosis. The cell cycle is a series of events that lead to cell division and replication. It consists of interphase and the meiotic phase. The main purpose of a cell cycle is to ensure that one cell divides into two genetically identical daughter cells, maintaining proper function and the integrity of genetic information. Mitosis, a part of the cell cycle, is a process where the nucleus of a cell divides, ensuring the equal distribution of chromosomes to each daughter cell. The cell cycle. Overview. The cell cycle is divided into two major phases: interphase and meiotic M phase. Interphase. This is the longest phase of the cell cycle, consisting of three stages. G1 phase. Gap 1. The cell grows in size, synthesizes proteins, and prepares for DNA replication. It is a phase of intense metabolic activity. The checkpoint here ensures that the cell is ready to proceed to the next phase. S phase. Synthesis. DNA replication occurs in this phase. Each chromosome is duplicated, resulting in two sister chromatids attached to the centromere. G2 phase. Gap 2. The cell continues to grow and prepares for mitosis. Additional proteins are synthesized, and the cell checks the integrity of the newly replicated DNA. Meiotic M phase. This phase is where cell division actually occurs. It includes both mitosis, nuclear division, and cytokinesis, cytoplasmic division. Mitosis is a complex process that ensures the division of a single nucleus into two genetically identical daughter nuclei. Mitosis. Detailed process. Mitosis is divided into several stages, each with its specific events. The stages of mitosis ensure that the sister chromatids of each chromosome are properly separated and distributed to the daughter cells. Prophase. Chromatin, a complex of DNA and proteins, condenses into visible chromosomes, each consisting of two sister chromatids. The nuclear envelope begins to break down. The meiotic spindle, made of microtubules, begins to form. The spindle fibers extend from the centrosomes, which are specialized regions of the cell that organize microtubules. The nucleolus disappears. Prometaphase. The nuclear envelope is completely disassembled. Microtubules from the spindle fibers attach to the centromeres of the chromosomes via a structure called the kinetochore. The chromosomes begin to move toward the center of the cell, aligning along an imaginary plane known as the metaphase plate. Metaphase. Chromosomes align at the metaphase plate in the center of the cell. Each chromosome is attached to microtubules from opposite poles of the cell, ensuring that sister chromatids will be pulled apart in the next phase. Anaphase. The sister chromatids are pulled apart toward opposite poles of the cell as the centromeres are split. This movement is facilitated by the shortening of the microtubules attached to the kinetochores. The cell elongates as microtubules not attached to chromosomes lengthen. Telophase. The separated chromatids, now called chromosomes, reach the opposite poles of the cell. A new nuclear envelope forms around each set of chromosomes. Chromosomes begin to condense back into chromatin, and the nucleolus reappears in each daughter nucleus. Cytokinesis. Although technically not part of mitosis, cytokinesis is the final step in cell division. It involves the division of the cytoplasm and other organelles between the two daughter cells. In animal cells, a contractile ring of actin filaments forms, leading to the pinching of the cell membrane cleavage furrow to separate the two daughter cells. In plant cells, a cell plate forms between the two daughter nuclei, which eventually leads to the formation of a new cell wall. Regulation of the cell cycle. The cell cycle is tightly regulated by checkpoints to ensure that cells only divide when they are ready. Key regulatory proteins include cyclins and cyclin-dependent kinases (CDKs). These proteins activate or deactivate the cell cycle machinery at specific checkpoints. G1 checkpoint restriction point. This checkpoint determines whether the cell should proceed to the S phase. If conditions are unfavorable, the cell enters a resting state called G0. G2 checkpoint ensures that DNA replication is complete and that there are no DNA errors before the cell enters mitosis. M checkpoint spindle checkpoint ensures that chromosomes are properly aligned at the metaphase plate before anaphase begins. If errors are detected, mitosis is halted to prevent aneuploidy, abnormal chromosome number. Importance of mitosis. Mitosis is essential for growth, repair, and asexual reproduction in multicellular organisms. It allows the organism to produce identical cells for tissue maintenance and regeneration. Errors in mitosis can lead to genetic disorders, such as cancer, where cells divide uncontrollably. Key concepts to focus on. Interphase, G1, S, G2 phases. DNA replication occurs in the S phase. Mitosis, prophase, prometaphase, metaphase, anaphase, telophase, chromosome alignment, sister chromatid separation. Cytokinesis, division of the cytoplasm and formation of two daughter cells. Regulation, cyclins, CDKs, and checkpoints ensure accurate division. Previous year questions. Question CSIRNET 2019. Which of the following occurs during the anaphase of mitosis? A, chromosomes align at the metaphase plate. B, sister chromatids are pulled apart toward opposite poles. C, the nuclear envelope reforms. D, the nucleolus disappears. Answer: B, sister chromatids are pulled apart toward opposite poles. Question CSIRNET 2020. In which phase of the cell cycle does DNA replication occur? A, G1 phase. B, S phase. C, G2 phase. D, M phase. Answer: B, S phase. Question CSIRNET 2018. Which of the following is true about meiotic division? A, the chromosome number in daughter cells is halved. B, daughter cells are genetically different from the parent cell. C, the process occurs in somatic cells. D, meiosis and mitosis are identical processes. Answer: C, the process occurs in somatic cells. This lecture provides an in-depth exploration of cell division and the cell cycle, focusing on mitosis and its stages. The information is designed to help you grasp key concepts in preparation for the CSIRNET JRF Life Sciences exam, ensuring you're well prepared for related questions. Let me know if you'd like further details or another topic. You said, meiosis, ChatGPT said. Meiosis, detailed process and key concepts. Meiosis is a type of cell division that reduces the chromosome number by half, resulting in 4 non-identical daughter cells, each with half the chromosome number of the original cell. It is essential for sexual reproduction, as it produces gametes, sperm and egg cells in animals and spores in plants. Meiosis consists of 2 consecutive divisions, meiosis I and meiosis II, each with distinct phases, and is crucial for maintaining genetic diversity through processes like crossing over and independent assortment. Overview of meiosis. Meiosis reduces the chromosome number by half and involves 2 rounds of division. Meiosis I, reductional division, where homologous chromosomes are separated. Meiosis II, equational division, where sister chromatids are separated, similar to mitosis. Before meiosis begins, the cell undergoes a preparatory phase called interphase, which includes G1 phase, cell grows and prepares for DNA replication. S phase, DNA replication occurs, resulting in chromosomes consisting of 2 sister chromatids. G2 phase, the cell prepares for meiosis by synthesizing proteins and other molecules. Meiosis I, reductional division prophase I. Chromosomes condense and become visible. Homologous chromosomes pair up in a process known as synapsis. Each pair of homologous chromosomes consists of 4 chromatids, forming a tetrad. Crossing over genetic recombination occurs between non-sister chromatids within a tetrad. This results in the exchange of genetic material, contributing to genetic diversity. The nuclear envelope breaks down, and spindle fibers begin to form. Metaphase I. The tetrads align at the metaphase plate. Homologous chromosomes are attached to spindle fibers from opposite poles, ensuring that each chromosome will be distributed to a different daughter cell. Anaphase I. Homologous chromosomes are separated and pulled to opposite poles. Each chromosome still consists of 2 sister chromatids. Unlike mitosis, sister chromatids are not separated at this stage. Instead, homologous chromosomes, which may carry different genetic variants, are divided, reducing the chromosome number by half. Telophase I. Chromosomes reach the opposite poles of the cell, and the nuclear envelope reforms around each set of chromosomes. Cytokinesis, division of the cytoplasm occurs, resulting in 2 daughter cells, each with half the chromosome number of the original cell haploid. Meiosis II, equational division. Meiosis II is similar to mitosis, but it involves the separation of sister chromatids rather than homologous chromosomes. Prophase II. Chromosomes condense, and a new spindle apparatus forms in each haploid cell. The nuclear envelope disintegrates. Metaphase II. Chromosomes align at the metaphase plate of each haploid cell. The spindle fibers attach to the kinetochores of the sister chromatids. Anaphase II. The sister chromatids are finally separated and pulled to opposite poles of the cells. Telophase II. Chromatids reach the poles and condense into chromatin. The nuclear envelope reforms around each set of chromosomes, and cytokinesis occurs, resulting in 4 non-identical haploid daughter cells. Key features of meiosis. Reduction in chromosome number. Meiosis reduces the chromosome number by half, ensuring that when 2 gametes fuse during fertilization, the resulting zygote has the correct diploid number of chromosomes. Genetic variation. Meiosis contributes to genetic diversity through crossing over occurs in prophase I, where homologous chromosomes exchange genetic material, leading to recombination of alleles. Independent assortment. During metaphase I, the homologous chromosomes align randomly, ensuring that the combination of chromosomes inherited by the daughter cells is unique. Haploid cells. The final outcome of meiosis is 4 genetically distinct haploid cells, gametes or spores, each with half the number of chromosomes as the original cell. Significance of meiosis. Sexual reproduction. Meiosis is vital for the production of gametes, sperm and egg cells in animals, pollen and ovules in plants. It ensures genetic variation through recombination and independent assortment, which is important for evolution. Prevention of chromosomal abnormalities. By reducing the chromosome number in gametes, meiosis ensures that the fusion of gametes during fertilization restores the diploid chromosome number, preventing chromosomal number doubling in every generation. Key concepts to focus on. Meiosis I. Homologous chromosomes are separated. Reductional division. Prophase I. Synapsis. Crossing over. Anaphase I. Homologous chromosomes are pulled apart. Meiosis II. Sister chromatids are separated. Equational division. Similar to mitosis but with haploid cells. Genetic diversity. Crossing over and independent assortment are critical for generating genetic variation. Haploid daughter cells. The end result is 4 genetically diverse haploid cells. Previous year questions. Question CSIRNET 2019. During which stage of meiosis does crossing over occur? A prophase I. Beat metaphase IC. Anaphase ID. Telophase I. Answer. A prophase I. Question CSIRNET 2020. Which of the following is a consequence of independent assortment during meiosis? A genetic recombination. Beat separation of homologous chromosome C. Formation of identical daughter cells D. Generation of genetic variation. Answer. D. Generation of genetic variation. Question CSIRNET 2018. The haploid number of chromosomes in the cells after meiosis is half of that in the diploid parent cell because of which of the following events? A DNA replication in S phase. Beat separation of homologous chromosomes during meiosis IC. Mitosis. Beat separation of sister chromatids during meiosis II. Answer. Beat separation of homologous chromosomes during meiosis I. This lecture provides a comprehensive overview of meiosis, detailing both the stages and the significance of the process in sexual reproduction and genetic diversity. By focusing on the key events, such as crossing over and independent assortment, you'll be well prepared for CSIRNet Life Sciences exam questions related to this topic. Let me know if you'd like another topic or further elaboration. You said. Mitosis and meiosis are regulation. ChatGPT said. Mitosis and meiosis, their regulation. Mitosis and meiosis are tightly regulated processes that ensure accurate cell division, proper chromosome segregation, and the maintenance of genetic stability in organisms. The regulation of these processes is crucial for preventing errors that can lead to diseases like cancer or genetic disorders. Both mitosis and meiosis are controlled by a complex network of checkpoints and regulatory proteins, including cyclins, cyclin-dependent kinases, CDKs, and other associated molecules. Regulation of mitosis. Mitosis is a process of nuclear division in somatic cells, resulting in 2 genetically identical daughter cells. The regulation of mitosis ensures that each cell division occurs correctly, with proper chromosome alignment, segregation, and cytokinesis. The key regulatory mechanisms include the cell cycle checkpoints, cyclins and CDKs, and spindle assembly checkpoints. Cyclins and cyclin-dependent kinases, CDKs. Cyclins are proteins whose levels fluctuate during the cell cycle. Cyclins bind to CDKs, cyclin-dependent kinases, forming complexes that drive the progression through different stages of the cell cycle. Cyclin D and CDK4/6 regulate the transition from the G1 phase to the S phase by promoting DNA replication. Cyclin D and CDK2 regulate the G1S transition. Cyclin A and CDK2 control the S phase and DNA replication. Cyclin B and CDK1, also known as CDC2, regulate the entry into mitosis M phase. This complex is essential for the transition from G2 to M phase and helps coordinate the steps of mitosis. Regulation of mitosis through checkpoints. G1 checkpoint restriction point before entering the S phase. The cell checks for DNA damage and whether the environment is favorable for DNA replication. If there is DNA damage, the cell will arrest at this point, allowing repair or apoptosis to occur. G2 checkpoint. Before mitosis begins, the cell checks for DNA replication errors and ensures that the DNA is undamaged. If any issues are found, the cell cycle is halted to allow repair mechanisms to fix the problems. M checkpoint. Spindle checkpoint. This checkpoint ensures that chromosomes are properly attached to the meiotic spindle before anaphase. If the chromosomes are not aligned properly at the metaphase plate or if spindle fibers are not attached correctly to kinetochores, the cell will not proceed to anaphase, preventing chromosome misregulation. Anaphase promoting complex APC-C. The APC-C is a ubiquitin ligase that targets regulatory proteins for degradation, ensuring the progression of mitosis. It marks securin and cyclin B for degradation, allowing sister chromatids to separate during anaphase. Spindle assembly checkpoint. SAC. The SAC ensures that the meiotic spindle is correctly assembled and the chromosomes are correctly aligned. It prevents the activation of APC-C until all chromosomes are properly attached to the spindle fibers. Regulation of meiosis. Meiosis consists of 2 rounds of cell division, meiosis I and meiosis II, and is essential for producing haploid gametes, sperm and eggs in animals and spores in plants. The regulation of meiosis is essential to maintain chromosome integrity and genetic diversity. The regulation involves similar checkpoints as mitosis, but there are also meiosis-specific mechanisms that govern recombination and segregation of chromosomes. Regulation by cyclins and CDKs. Like mitosis, cyclins and CDKs regulate key transitions in meiosis. The key difference in meiosis is that cyclins are involved in both the initiation of meiosis and the arresting of the cell cycle at specific stages. Cyclin B and CDK1 regulate the initiation of meiosis, particularly during prophase I, when homologous chromosomes undergo recombination. Cyclin A and CDK2 regulate progression through meiosis, especially during the transition from metaphase I to anaphase I. Regulation of meiosis I. Prophase I is the most complex phase in meiosis, as it involves homologous recombination, crossing over synapsis, pairing of homologous chromosomes, and the formation of the synaptonemal complex. The regulation of these processes ensures genetic diversity. SPO11. This protein introduces double-strand breaks in the DNA, which are necessary for homologous recombination. Checkpoint during prophase I. The checkpoint ensures that homologous chromosomes are properly paired and recombined before progressing to metaphase I. Meiosis I checkpoints. Synapsis checkpoint. This checkpoint ensures that homologous chromosomes are properly paired and recombined. If there is an error in synapsis or recombination, the cell may undergo apoptosis or arrest at the checkpoint. Spindle assembly checkpoint. Like mitosis, meiosis I has a spindle assembly checkpoint that ensures that the chromosomes are correctly aligned at the metaphase plate before anaphase I. This prevents errors such as aneuploidy. Meiosis II regulation. Meiosis II is similar to mitosis but involves the separation of sister chromatids rather than homologous chromosomes. Cyclin D and CDK1 play a crucial role in regulating the transition from metaphase II to anaphase II by ensuring that sister chromatids are correctly aligned before separation. The SAC spindle assembly checkpoint also plays a critical role in ensuring that the sister chromatids are attached to the spindle fibers and aligned before proceeding to anaphase II. Key differences in the regulation of mitosis and meiosis chromosome number. Mitosis results in 2 genetically identical diploid daughter cells, while meiosis results in 4 genetically diverse haploid cells. The regulatory mechanisms in meiosis ensure that homologous chromosomes are segregated in meiosis I and that sister chromatids are separated in meiosis II, maintaining the correct chromosome number in the gametes. Crossing over. Crossing over in meiosis adds genetic diversity by exchanging genetic material between homologous chromosomes. This occurs during prophase I of meiosis I and is tightly regulated by proteins such as SPO11. Spindle assembly checkpoint. Both mitosis and meiosis involve a spindle assembly checkpoint to ensure proper chromosome alignment. However, the SAC is more complex in meiosis, especially due to the need for homologous chromosome pairing and the prevention of non-disjunction during meiosis I. However, the SAC is more complex in meiosis, especially due to the need for homologous chromosome pairing and the prevention of non-disjunction during meiosis I. Cell cycle arrest. In meiosis, there are 2 key points where the cell cycle can arrest: during prophase I, where the cell can be arrested until recombination is completed, and during metaphase II, where the cell halts until the chromosomes are properly aligned. Previous year questions. Question CSIRNET 2019. Which of the following proteins are involved in regulating the G1S checkpoint in the cell cycle? Cyclin B and CDK1. Beat cyclin D and CDK4/6C, cyclin A and CDK2D, cyclin D and CDK2. Answer. Beat cyclin D and CDK4/6. Question CSIRNET 2020. In meiosis, what is the consequence of the spindle assembly checkpoint SAC failure during anaphase I? Homologous chromosomes segregate randomly. B. Sister chromatids segregate to opposite poles. C. There is an unequal segregation of chromosomes, leading to aneuploidy. D. Crossing over is inhibited. Answer. C. There is an unequal segregation of chromosomes, leading to aneuploidy. Question CSIRNET 2021. What role does CDK1 play in regulating mitosis? A. It prevents chromosome condensation. B. It regulates DNA replication during S phase. C. It activates cyclin B to trigger the onset of mitosis. D. It promotes cytokinesis. Answer. C. It activates cyclin B to trigger the onset of mitosis. This lecture has outlined the regulation of mitosis and meiosis, covering the key checkpoints, cyclins, and CDKs that control the progression of the cell cycle and ensure proper chromosome segregation. This information is crucial for understanding the molecular mechanisms behind cell division and for preparing for CSIRNet life sciences exams. You said. Steps in cell cycle. ChatGPT said. Steps in the cell cycle. The cell cycle is a series of events that lead to cell division and the production of two daughter cells. It consists of two major phases, interphase and M phase mitosis. The cell cycle is tightly regulated to ensure proper cell division, DNA replication, and the maintenance of genomic integrity. The primary steps in the cell cycle include 1. Interphase. Interphase is the longest phase of the cell cycle, during which the cell grows, carries out its normal functions, and prepares for cell division. Interphase is subdivided into three distinct phases: G1 phase, Gap1, S phase, synthesis, and G2 phase, Gap2. G1 phase, Gap1. This phase is characterized by the cell's growth and the synthesis of proteins necessary for DNA replication. The cell increases in size, produces RNA, and synthesizes proteins required for DNA replication. The G1 checkpoint, also known as the restriction point, checks for DNA damage and ensures that the environment is favorable for DNA replication. If conditions are unfavorable, the cell may enter a resting phase called G0, where it exits the cell cycle. If conditions are favorable, the cell moves into the S phase. S phase, synthesis. The S phase is where DNA replication occurs. Each chromosome is duplicated, resulting in two sister chromatids held together at the centromere. The replication ensures that each daughter cell will receive an identical set of chromosomes after division. DNA synthesis is regulated by various cyclins and CDKs, which control the progression of the cell through the S phase. G2 phase, Gap2. During the G2 phase, the cell continues to grow and prepare for mitosis. The cell synthesizes proteins required for cell division and checks for any DNA replication errors that may have occurred in the S phase. The G2 checkpoint ensures that the cell has fully replicated its DNA and that there are no errors before proceeding to mitosis. The transition from G1 to S phase and from G2 to M phase is regulated by cyclin CDK complexes, which ensure that the cell cycle progresses only when the appropriate conditions are met. 2. M phase, mitosis. Mitosis is the phase during which the cell divides into two genetically identical daughter cells. Mitosis is divided into several stages, each carefully regulated to ensure accurate chromosome segregation. Prophase. During prophase, the chromatin condenses into visible chromosomes, each consisting of two sister chromatids connected by a centromere. The nuclear envelope begins to break down, and the meiotic spindle, which is composed of microtubules, begins to form. The centrosomes organelles responsible for organizing microtubules start moving to opposite poles of the cell, and the spindle fibers begin to extend towards the chromosomes. Prometaphase. The nuclear envelope fully disassembles, and the spindle fibers attach to the kinetochores protein structures at the centromeres of chromosomes. Chromosomes begin to move towards the metaphase plate, the equatorial plane of the cell. Metaphase. In metaphase, the chromosomes align along the metaphase plate, ensuring that each sister chromatid is attached to spindle fibers from opposite poles. The metaphase checkpoint ensures that all chromosomes are correctly attached to the spindle before proceeding to anaphase. Anaphase. During anaphase, the sister chromatids are pulled apart towards opposite poles of the cell as the centromere splits. This separation is facilitated by the shortening of the spindle fibers attached to the chromatids, ensuring that each daughter cell receives an identical set of chromosomes. Telophase. Telophase is the final stage of mitosis, during which the separated chromatids condense back into chromatin. The nuclear envelope reforms around each set of chromosomes, creating two distinct nuclei within the cell. The spindle apparatus disassembles. Cytokinesis. Cytokinesis is the process of cytoplasmic division that occurs after mitosis. In animal cells, a contractile ring made of actin filaments forms, leading to the pinching of the cell membrane and the formation of two daughter cells. In plant cells, a new cell wall, the cell plate, forms between the two daughter cells. The successful completion of mitosis results in the formation of two genetically identical daughter cells. 3. Regulation of the cell cycle. The cell cycle is tightly regulated by a series of checkpoints that ensure proper progression through each phase. Key regulatory molecules include cyclins. These are proteins whose levels fluctuate during the cell cycle. They bind to cyclin-dependent kinases, CDKs, to regulate the progression through the cell cycle. CDKs, cyclin-dependent kinases are enzymes that, when activated by binding to cyclins, phosphorylate other proteins to drive the cell cycle forward. Cyclin CDK complexes. These complexes control transitions between cell cycle phases by phosphorylating target proteins that promote progression, e.g., cyclin B-CDK1 complex for mitosis. Checkpoints. The cell cycle includes checkpoints at G1, G2, and M phases, where the cell checks for DNA damage, replication errors, or incomplete division before progressing. Tumor suppressors, e.g., P53. Tumor suppressors such as P53 can halt the cell cycle in response to DNA damage, allowing for repair or triggering apoptosis if the damage is irreparable. Proto-oncogenes, e.g., cyclin D. Proto-oncogenes promote cell cycle progression. Mutations in proto-oncogenes can lead to uncontrolled cell division and cancer. Previous year questions. Question CSIRNET 2018. Which of the following is the correct sequence of events in the cell cycle? A. G1 right pointing arrow, S right pointing arrow, G2 right pointing arrow, M. B. S right pointing arrow, G1 right pointing arrow, G2 right pointing arrow, M. C. G1 right pointing arrow, G2 right pointing arrow, S right pointing arrow, M. D. M right pointing arrow, S right pointing arrow, G1 right pointing arrow, G2. Answer. A. G1 right pointing arrow, S right pointing arrow, G2 right pointing arrow, M. Question CSIRNET 2020. During which phase of the cell cycle is the DNA content doubled? A. G1 phase. B. S phase. C. G2 phase. D. M phase. Answer. B. S phase. Question CSIRNET 2021. Which protein complex is responsible for the initiation of mitosis? A. Cyclin DCDK4. B. Cyclin ECDK2. C. Cyclin BCDK1. D. Cyclin ACDK2. Answer. C. Cyclin BCDK1. This lecture provides a comprehensive overview of the steps in the cell cycle, from interphase through mitosis, with a focus on the key regulatory proteins and checkpoints involved. Understanding these processes is crucial for preparation for the CSIRNet life sciences exam. You said. Regulation and control of cell cycle. ChatGPT said. Regulation and control of the cell cycle. The cell cycle is a tightly regulated process that ensures the proper division and proliferation of cells. This regulation is crucial for maintaining cellular integrity and preventing diseases such as cancer. The cell cycle is controlled by a complex network of signaling pathways involving various proteins and checkpoints that ensure the fidelity of DNA replication, chromosome segregation, and cell division. Key regulators of the cell cycle. Cyclins. Cyclins are regulatory proteins that are synthesized and degraded at specific stages of the cell cycle. They bind to and activate cyclin-dependent kinases, CDKs, which are enzymes that drive the progression of the cell cycle. Cyclins exist in different types, each associated with specific phases of the cell cycle. Cyclin D acts during the G1 phase and is involved in the G1 to S phase transition. Cyclin E present during late G1 and early S phase, regulating the G1 S checkpoint. Cyclin A functions in the S and G2 phases, ensuring DNA replication and preparation for mitosis. Cyclin B present during G2 and M phases, necessary for the transition from G2 to M phase entry. Cyclin-dependent kinases, CDKs are enzymes that, when bound to cyclins, phosphorylate specific target proteins to trigger the progression of the cell cycle. The activity of CDKs is regulated by the availability of cyclins and their binding to cyclin CDK complexes. CDKs must be activated by cyclins to phosphorylate proteins involved in key events such as DNA replication, chromosome condensation, and spindle formation.